Hi, we're Nicole, Ricardo, and Mauricio, and we'll be talking about meiosis, specifically about the stages of meiosis, genetic variation, and non-disjunction. We'll start with the stages of meiosis. Meiosis consists of 4 distinct phases: prophase, metaphase, anaphase, and telophase. The 2 divisions of meiosis are separated into meiosis 1 and meiosis 2. During meiosis 1 in prophase 1, chromosomes become visible due to supercoiling. The paired and replicated homologous chromosomes are called tetrads or bevelins, which have 2 chromosomes and 4 chromatids. At this stage, non-sister chromatids may cross over at points called chiasmata. Centrioles, if present, migrate to opposite poles and spindle fibers start to form. The nucleolus and nuclear membrane disintegrate. During metaphase 1, homologous pairs or bivalents move together along the metaphase plate, which lies halfway between the 2 poles. The spindle fibers attach to the centromeres of each chromosome and start to pull gently in order to align the homologous chromosomes along the equatorial plane. Spindle fibers connect each centromere to one pole only. During anaphase 1, spindle microtubules shorten, pulling homologous chromosomes apart toward opposite poles. During telophase 1, the first meiotic division efficiently ends when the chromosomes arrive at the poles. The chromatids uncoil and a nuclear membrane then forms around each nucleus just formed. Telophase 1 is usually followed by cell division, which gives rise to 2 daughter cells with half the number of chromosomes as a parent cell, though each chromosome consists of a pair of sister chromatids. During meiosis 2, chromosomes with 2 sister chromatids each condense again. Centrioles, if present, migrate to opposite poles and spindle fibers start to form. The nucleolus and nuclear membrane disintegrate. During metaphase 2, the spindle fibers attach to the centromere of each pair of sister chromatids, which start to pull gently to align the sister chromatids at the equator. Spindle fibers connect each centromere to both poles. During anaphase 2, the centromeres are divided and the chromatids are moved to opposite poles by the spindle fibers. Once sister chromatids are separated, they are called chromosomes. During telophase 2, the chromosomes reach opposite poles and uncoil. This is followed by nuclear envelope formation and cytokinesis. Meiosis is now complete and ends up with 4 new daughter cells.
Now the genetic variation. It describes naturally occurring variations in the DNA sequences of individuals of the same species, allowing resilience and survival of a population when environmental conditions change. If this genetic variation is lost, the species is in danger of becoming extinct because of the lack of flexibility in their genetic makeup and cannot adapt to the changing conditions. So variation is considered beneficial for the protection it offers against. Genetic variation can be achieved by 2 methods. The first one is the crossover during prophase 1, in which homologous chromosomes come together and sections are exchanged between non-sister chromatids during synapsis. The other one happens because of the random orientation of the chromosomes during metaphase 1 and 2. The orientation of homologous chromosomes at the equatorial plate during metaphase 1 determines which pole each pair of sister chromatids will move to at anaphase 1.
Now we are going to talk about non-disjunction. During meiotic division, errors can occur. For example, during anaphase, the homologous chromosomes can fail to separate. This error is called non-disjunction, and it comes from the word failure, that means to separate. One of the most frequent forms of non-disjunction is trisomy 21. In this case, there are 3 copies of chromosome 21. If a child is born with such a genetic makeup, he or she may suffer from a number of health problems such as hearing loss, heart and vision disorders, growth retardation, and intellectual disability. The age of the parents can also influence chances of non-disjunction.
Thank you for your attention. We hope you enjoyed the video.